Good morning, everyone.
Good
Good morning.
morning.
Good
Good
morning.
morning. Good morning. Okay, just give me a minute. I'll just share the screen and then we can start. I think I shared the wrong screen. Yes. Okay. So, yesterday we discussed about transporters and channels. And now we will go into little more details of the types of transport and how do these transporters work. So if you see this image carefully, you should be able to see the basic difference that has been chalked out just using the figure. Okay, they have spoken about channels and transporters. Now, if you see the image A that is at the top, you will be able to see that they have shown you the differences of channels and transporters, wherein the first and foremost thing that should strike you is that they are showing that the transport can be without any transporter involved, wherein they have shown you simple diffusion. Okay, and they have also shown that with a transport protein involved, there can be transport happening in both the directions, that is, things moving out of the cell and things also entering inside the cell, where you can see that transporters, they have been shown to have specific sites where you will be able to see the molecules coming and binding. Okay, and you will be also seeing that how transporters can be highly specific for different molecules, because in one case they have shown you the transporter with a circular molecule, in the second case they have shown you a square shaped structure, right? They have also shown you the channels that are involved, and they have also shown you that transporters can be involved in both passive transport as well as active transport, because in case of active transport, you can see that there is supplementation of energy that is taking place, correct? Then in the second diagram, which is below, you are able to see how the electrochemical gradient matters a lot. Now, please understand that depending on what you are transporting, there could be an electrochemical gradient. For example, if it is ions that you're going to transport, no doubt it's a chemical agent, but because it has a charge, obviously now there is a charge-based gradient also, because of which you call it as electrochemical gradient. So many a times, most of the substances that are being transported, if they are charged, we refer to that as electrochemical gradient. In this image, you can see how normally the inner side of the membrane, the cytoplasmic end, is negatively charged. And you can see that the outside, because it is rich in positively charged ions, it has the positive charge. So how across the gradient the ions are being transported? Okay, so you need to understand that such a kind of a transport will be a diffusion process, wherein you don't have to force the transport, you don't have to supplement energy, because it is happening in the favor of the gradient. But when you go against the gradient, obviously you will have to involve ATP in it. So, whatever basic introduction that we have discussed is from Albert's chapter 11, and page numbers are mentioned over here. You can note that down for your reference. Any which ways, this PPT will also be passed on to you. Now, moving ahead to understand active transport process in more detail. First of all, you have to remember that a transporter substrate interaction is very similar to enzyme substrate interaction. Okay, so just like the enzyme has a catalytic site, an active site where the substrate needs to go and bind, and that site is highly specific for the substrate. Similarly, the transporter also has a binding site where the solute molecule has to go and bind. So the interaction looks very similar to what we call as the lock-in key mechanism. Remember that during this interaction, the transporter is not going to change the solute in any way, because in enzyme substrate interaction, we say that the enzyme will catalyze changes in the substrate to convert it into the product. That doesn't happen with the transporters, because transporters are expected to transport the solute across the membrane unchanged. Okay. Now you need to understand that when we say the transporter is specific for a solute, it can have multiple binding sites. Okay, now when I say multiple binding sites, for the same solute, a single transporter can have two binding sites, three binding sites, four binding sites, and so on. Which means that at a time, the transporter will be able to transport those many number of molecules of that solute. So it's not that a transporter always has to transport a single molecule of a solute at a time, because that's going to slow down the process, isn't it? So to favor time, even Cell has found this way wherein its transporter can have multiple binding sites for the same solute. Now, at times, it may also happen that the transporter has multiple binding sites, but for different solutes. So it may be specific for two solutes. It may be specific for three solutes because it has binding sites for those three different solutes. Okay, so you need to understand that transporter can have multiple binding sites either for the same solute or for different solute. Okay. Now, we discussed yesterday that when the solute goes and binds to the transporter undergoes structural changes. We said that it undergoes conformational changes, which helps it open in the opposite direction. But remember that these structural changes are not permanent. They are transient changes. They are happening only for that time period. Once the solute detaches from the transporter is now free to go back to its original conformation. Okay, so remember this, that the structural change that's happening to the transporter, that's not a permanent change. It is a totally reversible change. So you can see here they have shown you a similar kind of an example wherein you can see that it has two binding sites. Okay, and it is for the same solute, so which means that it can transport two molecules of that solute at a time. Now, until and unless both the binding sites are occupied, this transporter will not undergo a conformational change. So that's the requirement कि दोनों ही साइट यहां पर फिल होने चाहिए. जब तक दोनों साइट ऑक्यूपाई नहीं होंगे तब तक ये ट्रांसपोर्टर अपना स्ट्रक्चर चेंज करके दूसरी तरफ खुलने वाला नहीं है. Okay, the moment both solute molecules will bind, you see that the transporter has undergone a structural change and now it has opened in the opposite direction, and the solutes are free to detach from the transporter and move into the cytoplasm. Now, once both the solute molecules detach, only then this transporter will go back to its original state. Even if one solute is attached, it will still remain in this configuration where it opens towards the inside of the cell. Once both solute molecules leave, it is obviously going to be free because now there is nothing to keep it in that configuration. So once both solute molecules detach, it's going to revert back. Okay. Now, just like enzyme substrate interaction always involves kinetics, you also have transport kinetics. Please remember this. Whenever you talk about the rate of transport, there is this aspect of how fast the transport is taking place. Okay. So let us say that I have 100 molecules of the solute outside the cell and I have zero inside. Okay. So you need to understand that how fast the molecules will be taken inside the cell will first depend on how many transporters do I have. And a single transporter can transport how many molecules at a time. That will decide how fast the transport is going to take place. Now, let us say that the cell has only three transporters. ऐसा होता नहीं है, but I am just giving you an example. Let us say that the cell membrane has only three transporters, each of which transport one molecule at a time, which means that at a time the cell will be able to take in only 3 molecules inside the cell. Correct. Now, the rate at which this transport is going to take place is going to be slow. Correct. Why? Because I have 100 molecules outside, 0 molecules inside. There is a good concentration gradient. Obviously, this transport is going to be in the favor of concentration gradient, but the transporters involved are only 3. So I'm able to transport only 3 molecules at a time. So you need to understand that my transporters are saturated. There are so many molecules to be transported inside, and I have few transporters. So they are all saturated. So that's the maximum rate I can achieve, 3 molecules at a time, because I have only 3 transporters. Yes, if I had 10 transporters, then it would have been like 10 molecules at a time. Comparatively much faster rate, isn't it? If I would have had 25 transporters, then obviously the transport would be even more faster. But what I need to understand over here, that this transport will work in the favor of the gradient only till the time I have equal concentration of the solute inside and outside, which means 50 solute molecules inside, 50 solute molecules outside. Once this is attained, we have attained equilibrium. So now, if the cell still wants to take up more of those solute molecules, then the transport can take place. But what will it get converted into? Can anyone tell me what kind of transport will it now get converted into?
Active transport.
Very good. It will get converted into an active transport, because now you want to take 51st molecule inside, which makes the concentration of the solute higher inside the cell than outside. So you're going to work against the concentration gradient. Okay, so here also you have the concept of Vmax, you have the concept of Km. So Vmax is the rate at which the carrier can flip between 2 conformational states, which means how fast it is carrying out the transport. While Km is going to tell you the affinity that the transporter has for the solute. Now, please understand that if the Km values are higher, it is problematic, because Km always stands for affinity between the transporter and the solute. So can you tell me why is it problematic? We have briefly discussed this yesterday. If Km value is more, it indicates that it has a stronger affinity for the solute. What will happen in such circumstances?
Instead of solute getting released into cytoplasm, it will be bound. It will be more prone to be bound to the transporter.
Excellent. Excellent. So you need to understand that if the transporter is having a very high affinity for the solute, it will not leave the solute. And if it does not allow the solute to detach, are we successfully carrying out the transport? The answer is no. So we need to understand that surely we need to have some affinity for the solute so that the solute can come and attach to the transporters, but it shouldn't be so high that the transporter doesn't allow it to detach. Then the whole purpose of carrying out transport is going to be refuted. I hope you all are understanding this. If there is anything that you're not understanding, please stop me then and there. Okay? Everyone clear till this point?
Yes, sir.
So Km here also is concentration of solute where the rate of transport is half of Vmax. Very similar to what you learn in enzymology. And this is the graph which is also indicating the same to you. When it is simple diffusion, you normally see that simple diffusion is taking place across the membrane. Okay, there is no transporter. And because there is no transporter, there is no question of saturation. So it's going to be a straight line graph that you normally get in simple diffusion, because उसको किसी पे डिपेंड नहीं करना पड़ता ना, उसको रुकना नहीं पड़ता, अरे 3 ही ट्रांसपोर्टर है, वो एट अ टाइम 3 ही मॉलिक्यूल ट्रांसपोर्ट करता है, ऐसा कुछ भी नहीं है. सिंपल डिफ्यूजन में उसको सीधा मेम्ब्रेन क्रॉस करने का है, विदाउट हेल्प ऑफ एनी मेम्ब्रेन प्रोटीन. Okay, the simple diffusion happens much quicker and much faster, and that's the reason why we survive. वरना हमारा respiration सोचो ऐसे रेट में होता जहां पर ऑक्सीजन को बोला जाता है रुका अभी 3 ट्रांसपोर्टर है वो ऑक्सीजन अंदर लेगा फिर ही तू अंदर आ सकता है, तो अपना रेस्पिरेशन पता नहीं हम एक इनहेलेशन करते हैं और शायद 15 मिनट बाद एक्सहेलेशन करते हैं, ऐसा बिल्कुल नहीं होता, राइट? दैट इज ऑल बिकॉज सिंपल डिफ्यूजन हैपेंस एट अ मच मच फास्टर रेट, right? This is the beauty of how the entire thing has been designed. But in case of transporter, there is going to be a saturation. There's going to be a point wherein all the transporters, once they are occupied, the rate is going to become constant, correct? So that is what this graph is trying to tell you. Now transporters also have inhibitors. That's the best aspect of it, isn't it? Competitive inhibitors and non-competitive inhibitors are here also going to be seen. Competitive inhibitors are those which are very similar in terms of the structure and shape of the solute, and they have the ability to go and bind to the transporter, but they bind to the transporter in such a way that they don't detach from there. So what's going to happen in such scenarios? These inhibitors, they are competing with the solute to go and bind to the transporter. And once they bind to the transporter, they don't allow the transporter to interact with the solute. So transport has been halted. Similarly, we have non-competitive inhibitors also. These are those inhibitors which will not compete with solute because they are not similar in structure and shape to the solute, but they can surely have some other position on the transporter to go and bind to. And once they go and bind to that site, they will change the structural conformation of the transporter. Now, if the structure of the transporter is changed, especially if the site where the solute molecule used to come and bind, if that site structure is affected, then it is obvious that solute can no more go and bind to that site, which means that the transport is affected, correct? Now most of these inhibitors, they are reversible inhibitors, and let me also tell you that some of these inhibitors are also used as therapeutic agents whenever we want to act on some kind of physiological condition wherein the transporters have gone out of control. Some of these chemicals may be used to control that situation and to alleviate the symptoms. Okay, so you have to understand that there are inhibitors as well. Now what are active transport mechanisms? There are three major mechanisms by which active transport can be carried out. So what is that? One is making use of coupled transporters. The moment you hear the term coupled, you have to understand that probably two things here are being combined. So we are coupling the uphill transport of one solute with the downhill transport of the other across the membrane. Okay. Now, when you are doing this, you should always keep example of, such what you say exchange where proton is being taken inside, okay, ATP is generated, and you are trying to say get in something inside or throw something outside. So you can couple these two processes wherein protons are being pumped, and along with that you are trying to take something else inside or throw something else outside. So you're coupling the two-way transports with each other to balance it out. Several of the active transport work using ATP driven pumps, wherein existing ATP molecules are hydrolyzed, which is produced by respiration process, and you are then utilizing the energy released in that process to carry out transport. Maximum of mammalian cells have ATP driven pumps. Okay, coupled transporters यानी कि ATP वहीं बना के वही इस्तेमाल करो, ये जो कॉन्सेप्ट है ये बहुत ही कम है. But ATP driven pumps are the maximum metabolic energy maximum जो यूज होता है वो यही चीज में होता है मेम्ब्रेन ट्रांसपोर्ट करने में. और फिर अगर आप बैक्टीरिया की बात करो या आर्की बैक्टीरिया की बात करो तो वी वेरी वेल नो दैट देयर इज आल्सो कॉन्सेप्ट कॉल्ड लाइट ड्रिवन पंप्स, जहां पर बैक्टीरियो रोडोप्सिन इज एन एग्जांपल that they are deriving energy from sunlight, and whatever energy they derive from sunlight they produce ATP out of it, and they use that ATP for carrying out active transport. So these are the three mechanisms. एक, तुम टू वे एक्सचेंज करो जिसमें से एक एक्सचेंज प्रोटॉन्स का होगा उसमें तुम एटीपी जनरेट करो और उस एटीपी का इस्तेमाल करो, या तो रेस्पिरेशन में बने हुए एटीपी को तोड़ ताड़ के अपना ट्रांसपोर्ट का काम पूरा करो, और तीसरा सनलाइट में से एनर्जी डिराइव करके एक्टिव ट्रांसपोर्ट कैरी आउट करो. सो दीज आर द three methodologies using which you will see that cells carry out transport. So you can see here they've shown you coupled transport. So coupled transport looks very much like an antiport, wherein one stuff is being thrown out, another is being taken in. You have ATP driven pump, wherein you can see ATP is getting hydrolyzed, and you have light driven pump wherein energy is derived from sunlight. Okay, so you can see how, using figures you can normally depict uh, uh, any concept in cell biology. So keep this in mind, because if you have a written examination in the second term, you can always make use of these illustrations to explain your concept in a much better way. Okay, now, going to evolutionary relationships. First of all, let me tell you that when you look at transporters at the level of amino acid sequence, you will see that the transporters which are involved in carrying out passive transport or active transport, even though their mechanisms are different from each other, one needs ATP, another doesn't need ATP, still their amino acid sequences show strong similarities. It is also seen that the way they are folded, the structural conformations that they achieve, there might be differences, but there are a lot of similarities also in that design. So it is something very interesting, because this tells us probably how these transporters would have evolved eventually to get converted into transporters of active transport. Initially it would have been a passive transport, but eventually the cells would have realized that there's going to be a need for active transport, and probably the existing transporters themselves were modified a little and they got evolved to form transporters of active transport. Now, what you need to understand is that they've given you an example over here, that bacterial transporters, which derive ATP from proton gradient across the membrane, and the transporters which are involved in passive transport of glucose in animal cells. Look at that. We are talking about bacteria and we are talking about transporters in animal cells. We are saying that there is strong similarity between them. So this is also an indication of evolutionary relationship. Okay, so at the cell biology level also, several times when you look at the transporters, when you look at the cellular components, you realize that indeed the theory of evolution is very much true. We all come from the same ancestry. Okay, and this is not just at the molecular level, at the DNA level, or at the protein sequence level, but several times the design of the cellular structures also makes you believe this fact. So, in cell biology, at several point of time, right from division to other strategies and other structural features, you will realize that indeed we all come from the same ancestor, which is that unicellular archaebacteria, which came on Earth probably millions and millions of years ago. Going ahead, you know this concept from your lower classes. I'm very sure that these are the two terms which normally are spoken about: uniporters and coupled transporters. Uniporters are those transporters which transport solute only in a single direction. Matlab, if there is going to be movement inside the cell, this transporter is involved only in transporting things inside the cell. It's a one-way traffic. Okay, there's going to be no bidirectional movement over here. It's going to be only a unidirectional transport that's going to take place of solutes. That's the reason why you call them as uniporters, because of unidirectional transport. As far as coupled transporters are concerned, they are of two types: symporters, antiporters. Symporters are also called as co-transporters. Why co-transporters? Because please understand that symporters are those which transport two different molecules at the same time in the same direction. So if A and B are two different molecules, symporters will have binding sites for A and B. Only when A and B both bind to the transporter will it change its configuration and open to the other side. Which means that if A is absent and B is present, still B cannot be transported into the cell. And even vice versa, it is true. Okay? Which means that for the transport to take place, the presence of both the molecules is required. Now, this is the reason why you need to understand that the deficiency of one vitamin creates the deficiency of the other in our body. Many a times it so happens that our body really is deficient in a single vitamin, but it is co-transported with another vitamin. Okay, so because vitamin say 1 is deficient, vitamin 2 also will not be taken into the cell even though it is present, because both of these vitamins are transported into the cell by a symporter. So deficiency of one leads to the deficiency of the other. This is the reason why we say that, deficiency is one of the basic issues for most of the physiological conditions that we all suffer from. Because deficiencies can play a very big role in inviting several kinds of disorders into the system, and at the same time could make us prone to several kind of infections as well. While antiporters is an opposite concept. Antiporters also deal with transport of two molecules at the same time, but in opposite direction. So if A is going to move out, B is going to come in. So until and unless at both the sides, if A is inside and B is outside, until and unless both A and B bind to the transporter, bind to the antiporter, it will not begin with the transport process, because the transport in opposite direction or has to take place at the same time. And hence we call it as exchangers. Now, let me tell you, these also could lead to, physiological conditions if there is deficiency. Now, I'll say how. So, if there is deficiency of, say, one solute, which has to be taken inside the cell, so obviously another solute is going to will be thrown out of the cell during that process. So, अगर अंदर लेने वाले solute का deficiency है, तो जो सल्यूट बाहर फेंका जाना चाहिए वो बाहर फेंका ही नहीं जा रहा, वो अब सेल के अंदर अक्यूमुलेट हो रहा है. So in this case, deficiency of one leads to toxicity of the other. See, in symporters, it was deficiency of one leading to the deficiency of the other. Here we are saying deficiency of one leads to toxicity of the other. Is it making sense to all of you? Are all of you understanding this two concept here, symporters and
Yes, sir.
Yes, sir.
Yes, sir.
Okay, fine. So तुमको इसके ऊपर क्वेश्चंस आ सकते हैं जहां पर तुमको केस स्टडी की तरह क्वेश्चन दिया जाएगा, and you will have to try and understand that what is going wrong in the transport process. So when you give test, you will see such kind of questions. So read the questions carefully and then come to a conclusion on what is going wrong. Okay. Now, please understand that many a times it happens, most of the times, even in lactose permease, you would have seen that in TY biochemistry, you were taught about lactose permease, and you were told that it is responsible for secondary active transport of lactose, if you remember. This is because in case of secondary active transport of lactose, first what we do is we create an electrochemical gradient of protons, and using that electrochemical gradient, we generate energy. That energy is then used to transport lactose into the cell. So it is like you're harvesting or you're harnessing energy which is present because of electrochemical gradient of one solute to transport lactose. So this is also one of the very common ways, and it's example of coupled transporters. Mostly you will see that such transporters are very, very common in bacterial cells. but also in mammalian cells, you will find few transports taking place in this manner. Okay. Now, sodium is usually co-transported in animal cells. That there is a reason behind this. Please understand that. Sodium concentration inside the cell is always kept at bare minimum. Okay, so whenever there is sodium flux, what is flux? Please understand these three terms now. Flux means movement, it could be in either direction. Influx means inside the cell, something is moving inside the cell, that's what you call as influx. And what is efflux? Efflux is something is being thrown out of the cell. So three very important terminologies in transport: flux, influx, efflux. Flux is movement in either direction. Influx is movement inside the cell. Efflux is movement out of the cell. Now, sodium ions normally are co-transported in animal cells. What does this mean? So when sodium ions are in excess outside, हमने कल देखा था 145 मिलीमोलर बाहर था और अंदर हम नॉर्मली 5 to 15 मिलीमोलर्स का कॉन्सेंट्रेशन मेंटेन कर रहे थे सोडियम का. प्लीज याद रखो, कॉन्सेंट्रेशन ग्रेडियंट होने की वजह से सोडियम सेल के अंदर आने वाला ही है. बट ये सेल का अब स्ट्रैटेजी होना चाहिए कि कैसे इनफ्लक्स जो हो रहा है सोडियम का, उसके बावजूद सोडियम के कॉन्सेंट्रेशन को सेल के अंदर मेंटेन करके रखा जा सके एट अ लोअर लेवल. अब सेल का स्ट्रैटेजी क्या है? मैक्सिमम एंटीपोर्ट में सोडियम को बाहर फेंका जाता है और उसके बदले में जो चीज चाहिए वो अंदर लिया जाता है. ओके, कई बार you will see that the cells will purposefully take sodium inside the cell so that antiport can be carried out, because sodium will be pumped out and the other component will be taken in, and you can also generate ATP in this process. Instead of proton, these will be sodium pumps which are going to be functioning over here. So antiports maximum that you see in animal cell, you'll be seeing an example also. Sodium potassium pump is one of these examples wherein you see that sodium is thrown out and potassium is taken in. So ATP driven carriers, they are primary active transport because they directly hydrolyze ATP and they carry out the transport. Ion driven carriers are wherein you first create a gradient, generate ATP and use that ATP for transport of your solute. That's secondary active transport. जैसे मैंने lactose का example already आपको दिया हुआ है. So this image again shows you uniport, symport and antiport. I think the image very clearly depicts that uniport is a single direction transport, a unidirectional transport. Symport is wherein two molecules are being transported in the same direction at the same site. So you can see that it has two binding sites for two different molecules. While antiport is wherein one molecule moves in and other molecule moves out. Again, you can see that there are two binding sites for the same. Now, intestinal cells, kidney epithelial cells, these are rich with transporters. क्योंकि इनका काम ही है ट्रांसपोर्ट में इन्वॉल्व होना. याद रखना, इंटेस्टिनल एपिथेलियम में से थिंग्स हैव टू मूव आउट एंड पास इनटू द ब्लड. They need to have efficient transporters over there. Kidney has a lot of work in filtration, urine formation, reabsorption. So in this, obviously transporters have to be present. Otherwise things are not going to work efficiently. Now, as I told you that sodium can be involved in antiport, sodium may also be involved in symport, wherein some symporters take sodium ion and sugars or amino acids together. So one space is occupied by sodium ion and other space is occupied by, say, a specific sugar or a specific amino acid, depending on what the transporter is for. And then the sodium is, taken along with the sugar inside the cell. Okay. So any which ways, this is a favorable process for a basic reason that सोडियम इज बीइंग ट्रांसपोर्टेड अक्रॉस द इलेक्ट्रोकेमिकल ग्रेडियंट. बाहर सोडियम का कॉन्सेंट्रेशन ज्यादा है और अंदर कम है, तो इस इलेक्ट्रोकेमिकल ग्रेडियंट के लिए ये फेवरेबल ट्रांसपोर्ट है. तो सोडियम के साथ आप एक तरीके से सोल्यूट को अंदर खींच रहे हो, यू आर ड्रैगिंग इट इनसाइड. Okay, and obviously your question would be that now if we are taking sodium inside, we are increasing the internal concentration of sodium. How is that managed? So remember, cell always will throw that excess sodium outside using an antiport mechanism, so that that lower concentration of sodium is always maintained. तभी तुम और solute को अंदर खींच सकते हो against, not against, but in favor of the electrochemical gradient. So it always tries to maintain lower sodium concentration inside. So भले ही सिम्पोर्ट के साथ सोडियम अंदर आया, जो भी सोडियम अंदर आया है उसको एंटीपोर्ट के जरिए वापस बाहर फेंक दिया जाएगा, ताकि सोडियम का कॉन्सेंट्रेशन अंदर लो ही रहे हमेशा. तभी ही तुम सोल्यूट को अंदर खींच सकते हो along with sodium. Okay, so greater this gradient is, greater this differences of sodium ion concentration, faster will be your solute transport. And that's the reason why the cell never takes the risk of accumulating sodium inside. The cell starts accumulating sodium inside, gone, your organs are going to fail. Okay, so you have to understand that sodium has to be given a chance to move out quickly, so that that concentration gradient is always maintained. Now, in bacteria, yeast, and several membrane-bound organelles, they may not depend on sodium. They may depend on proton instead for solute transport. So instead of sodium pump, they will have proton pump. So lactose permease is the best example. They create a gradient of protons and then transport lactose. Okay, that's very familiar protein that is studied in case of E. coli. So here you will be able to see the functioning of permease, how lactose permease works. Lactose permease is a transmembrane protein wherein it has 12 helices. Imagine this is like your stitching of the protein that we spoke about yesterday, 12 alpha helices on the membrane. Okay, and their movement is very, smooth movement that you see in the membrane, because it just tilts and slides to open and close. So if you see the structure over here, this is the 12 helices that you see. There is a spot for the proton and lactose to come and bind because it's a co-transport. And you can see that once both of them attach, how they just slide a little to open in the opposite direction so that this can be transported. But अब ये जितना आसान दिखता है ना स्टूडेंट्स, ये उतना आसान है नहीं. ओके, ये क्यों आसान नहीं है, या why do I say it's a complicated process, is that it has specific sites of binding, students. These numbers that you see are indicating the amino acids where they come and bind. Okay, so the first thing to come and bind to this structure is going to be a proton, which binds at the right hand side helices section. Once this binds, it makes the structure, in a way ready to bind to two lactose molecules. So you can see that lactose comes and binds over here. And once lactose binds, it immediately undergoes conformational change, because this binding, changes the way amino acids interact with each other. So as the interaction changes, in दोनों के बीच में जो interaction था वो टूट चुका है. Now the interaction has shifted to the next two amino acids, thereby changing the conformation. Now it opens in the inside of the cell. So both the proton and the lactose can leave. So first the lactose will leave and then the proton will leave. And then now this interaction could be restored to its original type, and this transporter will open outside. This is how the entire transport is supposed to take place. So हमको जितना आसान ये पूरा प्रोसेस लगता है, उतना आसान ये है नहीं. Okay, it's a very complicated process. The people who study it take like years together to understand how a transporter is functioning, what is the mechanism by which it is functioning, which are the amino acids which have the most important role there, and if you are changing these amino acids, probably then you cannot expect this transport to take place in this fashion, because those amino acids are extremely important and they are responsible for the kind of transport that is taking place right now. So even if that single amino acid is changed, gone, the entire transport is going to fall apart. So that is something that we all have to understand and remember. There are several transporters which work only for regulating cytosolic pH. Can you tell me why is the pH of the cytoplasm so significant? Can anyone tell me why is it important for maintaining cytoplasmic pH? Anyone?
Sir, because we have active sites there where other transporters. So if we change the pH, then the enzymatic activity also will change, like for the affinity and all.
Okay. Anyone else?
So pH means it will be directly related to the ionic nature of cytoplasm. So means if it is low pH means high H plus concentration and like that.
Okay. So, first and foremost, you need to understand that cytoplasm is the site for maximum metabolic activities. Okay. So, if the cytoplasmic pH drifts from what it is, normally, all cells will try to maintain their cytoplasmic pH close to neutral. Whether it's bacteria, whether it's animal cell, plant cell, mammalian cell, doesn't matter. The cytoplasmic pH is always close to 7. Okay. Now, this helps these, cells to carry out their metabolic activities very normally. As if the pH drifts towards extreme, the enzymes, the proteins, all will get affected in terms of their structural conformation. The activity of those proteins and enzyme is going to be hindered. And most importantly, nutrient acquisition becomes a problem. Acquisition means acquiring nutrients. Why is it so? Because when pH changes, as Ritika very correctly mentioned, ionization takes place. So even nutrients get ionized. And when nutrients get ionized at extreme pH values, it becomes impossible to utilize them. So it is important for us to understand that cytoplasmic pH has to be maintained close to 7.2. That's the optimal pH for all metabolic activities. Yes, some organelles may have lower pH, like lysosomes, it maintains its pH at 5. But majority of the other organelles will have a pH which is close to 7. And cytoplasm always maintained at around 7.2. Please understand that several transporters will work only to maintain this pH. बाहर भले ही कोई भी काइंड का पीएच क्यों ना हो जाने दो, बाहर कंडीशन कितना भी एसिडिक, कितना भी अल्कलाइन क्यों ना हो जाने दो, इवन द एक्सट्रीमोफाइल्स विल ट्राई देयर लेवल बेस्ट not to let the cytoplasmic pH get affected. For this, if you take example of an acidic environment in which the cell is, it is obvious that there are a lot of protons outside because it's an acidic environment, and because of electrochemical gradient, these protons will get into the cell. Now that's going to acidify the cytoplasm, which is dangerous. So then what will the cells do? They will pump out excess of the H plus protein, or rather, sorry, the H plus ions, by exchanging it for sodium ions. So pump H plus out, take Na plus in. Okay. You can also have other strategies wherein you try to neutralize it. Okay. That strategies are also available. It depends on the cell. Now, because you're doing this exchange with sodium, sodium ion concentration will increase inside. So even that has to be handled. How will you handle that? Either you use an antiport to pump those sodiums out later, or you can even neutralize those sodium ions by combining them with bicarbonate ions. Okay, HCO3 minus ions. So even that is possible. But at that point of time, taking in sodium becomes important to pump out the excess of protons, that becomes very, very significant. Okay, please understand that. This is the reason why cells normally also have this habit of having buffering agents inside them, which are mostly proteins or peptides or even some amino acids. Remember that your blood also has excellent buffering system. Okay, and it's not just salts. Obviously, there are several salts which function there as buffering agents, but you would be shocked to know that your hemoglobin, which carries oxygen, it itself is a buffer. It's a protein, but it's a buffer. Why do we need buffer in blood? We need buffer in blood because remember that the waste product that we produce in respiration is carbon dioxide. That carbon dioxide combines with water in your blood. When CO2 is coming out of the cells into the blood, it's combining with water in your blood. And what is it producing? Water and carbon dioxide is carbonic acid. This carbonic acid is going to acidify your blood, which is obviously disastrous. So we need a buffering agent there. So we already have sodium, potassium, bicarbonate salts as the buffering agent over there. But along with that, hemoglobin itself is a very good buffering agent that functions there. Remember that. Now, here is where, I would want you to understand the process of respiration. So I think I will have to show you this image. But before we get into this, I want you to understand this simple story of respiration. Please understand that हीमोग्लोबिन की पक्की गर्लफ्रेंड है ऑक्सीजन. Hemoglobin loves oxygen, madly in love with oxygen. The affinity that hemoglobin has for oxygen is zillion times more than it has affinity for anything else. It loves oxygen to that level. So hemoglobin and oxygen combine to form this jodi which is called oxyhemoglobin. I'm sure you all have learnt of this in your lower classes. And that's how they travel around your entire system. But when it reaches tissues in cells, oxygen decides to dump hemoglobin. She thinks, I don't want to be with you anymore. So oxygen dumps hemoglobin and enters into the cells because that's where it is needed for electron transport chain. So oxygen dumps hemoglobin and it goes into the cells. Now hemoglobin is very depressed. It's very unhappy. Okay. Now it is unhappy, but then it sees something coming out of the cell as oxygen goes inside, and that something is carbon dioxide. Okay. Now, as it sees carbon dioxide, it's not focusing on C. Remember what hemoglobin loves: oxygen. So hemoglobin thinks O2 नहीं तो क्या हुआ, CO2 तो है, and decides to combine with carbon dioxide. That's how you get carb amino hemoglobin, which is a combination of carbon dioxide and hemoglobin. Some part of your carbon dioxide is carried in that format for, exhalation purpose. Never CO2 and oxygen travel as free molecules in your blood. Remember this. Never ever. But what happens in this story further is governed by three laws. There is Bohr's law, there is Haldane effect, there is Hamburger phenomena, which is called as chloride shift. But to understand this, we will have to obviously go through all the three laws. Only then we can come to this exchanger concept, which we will obviously discuss on Monday, because even if we start, we will not finish it today. Those are three heavy concepts which we will have to write and understand. So I will be putting up my whiteboard and will explain you the reactions, but we will do that in the next lecture. So these three phenomena we will discuss in our next class. If you have any questions till this point, please feel free to ask. We can discuss that. or we would be ending the session for today. Also, next Saturday, the date next Saturday is 5th of Feb. Whatever we discuss till मंडे, मंडे के लेक्चर तक जो भी हमारा डिस्कशन होगा, वहां तक मिलाकर you are going to write a test. It's not going to be MCQ, it's going to be return test, because now you have to prepare yourself for that. Okay, so it's going to be a written examination. I will put the questions in the classroom, Google Classroom, and you can take your time, maybe 2 days, to write the answers to those questions. And any which ways, next week onwards, you will be coming to college. So you will submit those papers to me, even though the paper is on Classroom. You will submit those papers to me manually and not virtually on the Classroom. Okay, so I can correct those and give it back to you. So whatever we cover till Monday, you are going to prepare for that. Is it okay with all of you?
Yes, sir.
Okay. So I will post this presentation just now itself so that you can start preparing if you wish to. Where is the classroom? Okay, it's taking time to load. So I will post it there already. The recordings for yesterday's lecture also has been posted. Today's lecture also I will post. So if during studies you have any difficulties, you can go back to the recording and revisit it. Okay, and if you have any questions, please feel free to ask me anytime. Okay, see you soon in college and